Get started. So thanks everyone for joining us this evening for the AHPBA Clinical Trials webinar series. Today is going to be the first of our four-part webinar series focusing on introduction to clinical trials, really highlighting design and feasibility. My name is Akil Chawla. I'm a surgical oncologist at Northwestern University. It's my pleasure to co-moderate with Dr. Sepida Gulami from Northwell Health. As I mentioned, today is the first part of our four-part webinar series, and this is the third year that the AHPBA Clinical Trials Committee has really put out this exciting webinar series focusing on educating both trainees and faculty in the clinical trial design and implementation aspects that are so important for surgeons within our field. Today we have two speakers. Each will have 45 minutes, inclusive of questions opened to attendees at the end of each talk. Certainly want to open up any additional questions and suggestions for future trial concepts and future, it's not trial concepts, but rather topic concepts that we want to introduce within our webinar series. And I'll turn it over to Dr. Gulami now to introduce our first speaker. Thanks, everyone.
Thank you, Akil, for this great introduction. I think just a few more items that we thought we're bringing back by popular demand is that we will be handing out certificates at the annual AHPBA meeting if you attend all four sessions, and we'll complete the survey that will include some questions from the speakers that we usually get. These are going to be pretty brief questionnaires, but we really want to make sure that you not only attend, but also really have a good understanding of the concepts and can use the certificate to, put on your CVs, and again, for the trainees or even the early faculties. And we got some great feedback on that. And then just a couple of housekeeping rules is that there is a chat. If you have any particular questions, please use the chat feature. And for the Q&As, we really want this to be an interactive session, and want to make sure that you really understand, and it's just less lecture, although there is going to be obviously some lectures in it. But I think just from the past 2 years, the discussions were really crucial to us learning from our speakers, who were really honored to have today. So without further ado, I'm going to introduce our first speaker, a dear friend and colleague, Dr. Chris O'Callaghan, who has a really interesting background with his doctorate in veterinary medicine, having a master's and a PhD degree in epidemiology. And really, I just learned that really he did some research in Nairobi, Kenya, and also some work in Zimbabwe. I met Dr. O'Callaghan really through my work in clinical trials and working with a medical oncologist there through the Canadian Cancer Trials Group. So Dr. O'Callaghan's really the senior investigator and really oversees the gastrointestinal and brain side committees there. And I really had an incredible pleasure to not only work with Chris, but really also learn from Chris. He's just a wealth of knowledge. So it is my great pleasure to have you here and really lead us into really what can you do and teach us about how do you lead a successful clinical trials before you even entertain in designing one.
Thanks very much, Seth. I'm going to try and share my screen here, so bear with me if it's a problem. Can everybody see that now as a single slide?
Yep, that's perfect.
That's great. So I'm going to try and keep this a little bit high level and also have a little bit of levity in it. As Seth has said, I've been doing clinical trials for quite some time now. It scares me sometimes to think how long, but the Canadian Cancer Trials Group, I'm going to give you a little bit of background about who we are. For those of you who are familiar with the US cooperative group structure, you guys have 5 academic cancer cooperative trial groups within the United States. We have only one in Canada, and we are the national group. And so we are the only group that actually does adult cooperative group trials in cancer. We're based at Queen's University in Kingston, and we're a kind of a one-stop shop. So all of the elements or all of the skills that are required, or the components even, that go into designing, running, and analyzing clinical trials are resident within our group. And we, we have done for the last 45 years quite an extensive amount of clinical trial research. We're based, as I said, as at Queen's. We have 85 institutions across Canada who are our members. But one of the things that I will allude to in my talk today is that clinical trials work is really no longer possible in a single country. Even a country as large as the United States, with the population base that you have, it's not unusual for larger trials to be looking for international partners. And we, for quite some time, have joined that trend and have been working with 40 different countries around the world, putting lots of patients on, doing lots of trials. At any one point in time, we have about 150 trials that are actually ongoing. Our major sponsor is the Canadian Cancer Society, and I will speak a little bit more about their role when it comes to how we all fund the clinical research that we're doing. And of course, evolving in keeping with the emerging technology and focus, we have been expanding and continue to expand our correlative research facilities and biobanking. So as I say, we've been doing this for about 40 years. My personal involvement has been 25 years. And so everything I'm going to tell you today is really from what I would call myself as an academic clinical trialist. And so I'm going to be slightly provocative and say that, from my perspective, a successful clinical trial is well designed, conducted in a timely manner. And I've emphasized that because I think that is one of the areas that we as clinical trialists don't often consider, or we expect. We have high expectations that, of course, we'll be able to do our research in a timely way. And when we fall into difficulties in meeting a timely outcome or a desire for a timely outcome, we often start to question ourselves. We need to put high quality. We need to produce high quality data. We obviously are going to stringently analyze and then fully and transparently report. But it's not necessarily a positive trial. Of the trials that we've done, the 500 trials, it's always nice to have a positive trial. I had a trial 2 weeks ago that read out where I have to be honest, I was expecting a negative result. A positive result is a wonderful thing. But for many of our trials where we reach the conclusion that we did not demonstrate what we were hoping to, that does not mean that we haven't made an important contribution to the question, or importantly, to potentially even changing or tempering our standards of care or practice. And so the corollary to that is, well, a successful trial is one that gives us an answer. A failed trial, in my opinion, is one that doesn't give us the answer. And there are all sorts of methodological problems that might be involved in that. We could have bias. We could have an uninterpretable or an inconclusive result. We could end up with an underpowered trial, a flawed trial. We've even had circumstances, it's rare anymore, but there's always the possibility that fraudulent activities can contaminate a trial. But by far and away, the number one problem that I have seen in my experience is a well-designed trial that simply fails to recruit patients. And I would consider this to be the number one challenge for academic clinical trialists, regardless of your discipline, whether you're a surgeon, radiation oncologist, medical oncologist. These trials are the true failure in that they waste the time, effort, resources, and importantly, they're an opportunity cost. If you imagine that you spend considerable amount of time trying to conduct a clinical trial that ultimately fails simply by not recruiting adequately, you could have been doing another trial. You could have been answering another question. And now I'm going to say it should be simple, right? I mean, we design a clinical trial. You guys are going to hear more about what goes into the thought process of designing the trial, but it's not rocket science. There's lots of good information out there. There's lots of experience. So surely this is something that we can do as an enterprise. We recruit patients to the trial. We collect data from those patients, and hopefully we're collecting biospecimens now. And then we analyze and answer the question. And I would argue to you that number 1, 3, and 4 are something that an organization like ourselves, or the opportunity for you to learn and get information about how to do clinical trials, is available. And smart people, careful planning, peer review of our proposals, monitoring of the trials as they're going. This is the science behind clinical research. And I think that is pretty well established. My concern, as I'm going to talk to you about today, is the intangible. The patients who are not going on the trial, perhaps due to patient preference, perhaps due to investigator preference, perhaps simply due to red tape and costs associated with trials. And I'll speak a little bit more about those. There is the sort of one area of crossover, and I'm just putting it out there as a reminder to ourselves that if you design a trial where your eligibility criteria are so restrictive and so numerous that you struggle to find any patients that meet them, you can thwart yourself in that method, or that way, by being unable to recruit for that reason. But there's certainly one absolute truth, which is that a clinical trial must recruit patients. Larger trials require more patients, more collaborators, more effort. And the one thing that I have observed is that we as an organization, or let's say my own organization for sure, but other organizations also suffer from this. We rarely are able to predict what we are going to actually accomplish on a clinical trial. And I want to spend a little bit of time about that. This is airing the Canadian Cancer Trials Group dirty laundry. And I've called it the good, the bad, and the ugly. And each of these graphs represents the accrual on a trial. So if you look at the graph in the top left, the solid curve is is the actual cumulative recruitment that was achieved on the trial, and the dotted curve is what we as an organization had predicted we would be able to do. And so the top left trial, I'm calling the superhero trial. This is the scenario where you start your trial and you are overwhelmed with the recruitment on the trial. It's exciting, it's interesting. Patients want to go on the trial, investigators want to participate on the trial. You open it up thinking, well, we'll put we'll have 5 or 6 centers that open the trial, and suddenly you have 20 institutions that want to join. That is a wonderful scenario to be in. I think it's happened to me twice in my career. So it can be achieved. It's a little bit more rare. I love this one. This is the hockey stick effect trial. And this is the scenario where you're struggling to get the trial up and running, but once you actually do, it actually recruits extremely well. And this is something that I've seen in the scenario where we have perhaps difficulties for the sites in activating a study, but once you've got the sites up and running, the trial recruits very well. The bottom 4 are unfortunately the trials that we often see. This particular trial I know very well. This was a positive trial. This trial read out, it's a Lancet Oncology publication about palliative radiation in patients with liver cancer, but it was a quicksand trial. We started out fairly well. Recruitment was slower than we predicted, but just fell off over time. And in the end of the day, we were very lucky to simply be able to get the trial done. The late to the party pattern, where you achieve your recruitment eventually, this is a bit like the one above it, but for a variety of reasons, you're delayed in getting the trial started. And that delay can sometimes be also fatal to the trial. But the worst trials are the marathon trials. The trial in the bottom left here is one where we eventually had to petition our Data Safety Monitoring Committee to allow us to do an early analysis, and frankly, what will eventually be an underpowered analysis simply because we couldn't recruit the patients that we thought we could. And I think there are a number of reasons that trials do well, but the number one factor that comes over and over to me again is that it's an interesting trial. And this is often a little bit difficult. Everybody who is creating their own trial, and Sepida will speak to this as well. If it's your baby, you're interested in it, but are your colleagues interested in it? Can you engage a community of similar, like-minded investigators who will agree to put patients on your trial? Will their institutions, more importantly, support them? And are they capable? In other words, are they perhaps adequately staffed? Is there a clinical trials infrastructure? And I know that in the surgical community, this is one of the things that I hear from my colleagues, is that they feel that our medical oncology and radiation oncology colleagues are a little bit ahead of them in that they have access to or have created infrastructures for conducting clinical trials. But of course, in the end of the day, it has to be something that the patient is interested in participating in. And the goal is rapid activation of the study and timely accrual. And you always have to expect that there's this attrition that happens where if you're trying to figure out, well, how many patients do I really think I'm going to be able to enroll on my trial? Well, obviously you can figure out what the number of incident cases are. You then maybe have a pretty good idea of being able to figure out, based on what your eligibility criteria are or your target population, you can figure out what the attrition will be between those incident cases and the eligible patients. But the real question is going to be, how many of those eligible patients, when approached by you or your colleagues, will agree to participate in the trial? And one of the things that has happened, I've alluded to this about the idea of having international collaborators, is casting a broader net. Sepida and I are working on a trial right now that's a collaboration between Canada and the United States. But we have done trials where, we've needed to have a sample size of somewhere around 2,000 to 3,000 patients. I'm thinking about adjuvant breast cancer trials, adjuvant lung cancer trials. When you're looking at trials of that magnitude, you really need to be casting a very wide invitation to collaborators. We in Canada, with a population base now of somewhere around 35 million people, rarely are able to do a definitive phase 3 trial within country in a timely way. And in part, that is because the rates of accrual that we're able to achieve are now a fraction of what they once were. So we look elsewhere. We engage with other groups. We are collaborators with our colleagues in the United States, but we're collaborators internationally. And that's one of the ways that we as an organization have tried to overcome this issue of attrition between incident cases and patients who are willing and consent to go on trials. There are a number of models. Our colleagues in pharma often use a model that I would call the international model, which is they go around the world looking for the top recruiting sites, and they sign up individual institutions, perhaps in relatively small numbers in any given country. For us in the academic world, that's an incredibly expensive proposition. It is by far and away the most expensive way on a per patient basis or cost per patient basis to run a trial. So it's not something that in our world we look to. We prefer that previous model where we're engaging other academic cooperative groups like ourselves. So what's the question? What makes a trial interesting? And I've said, well, I think it has to be a relevant question. It's something that's not expected to be superseded. If you're asking a question that is likely to be answered before you've done the trial, then it's not by definition an interesting trial. If you have promising data from earlier stage trials, new novel drugs or treatments, and I know I'm speaking to an audience of surgeons here, but in the world of medical oncology, it's absolutely true to say that it's easier to sell a trial with a novel new drug than repurposing an existing drug. Simple trials are more attractive. Complexity scares patients. It also increases cost. And frankly, if you have a scenario where there are limited therapeutic options, for example, end-stage settings, those are the trials that patients themselves are often looking for. And patients need to feel that there is at least a perceived good risk-benefit ratio to them. The trial needs to be something, as I've said, that's not already planned or in progress. If you're duplicating a trial idea or someone else has started a trial ahead of you, it may be something that you need to rethink. And when I say well-funded and resourced, even in the world of academic clinical trials, and even in a scenario where you are speaking to your colleagues and appealing to them to support your trial idea or an idea that you bring forward, it is completely naive to expect that they would do that without having adequate resourcing to do so. And that is becoming increasingly challenging for us. And of course, all of these, none of these is a fatal failure. They're like rungs in a ladder. They're not all absolutely essential, but the more that you have, the easier it is to get the trial done. As an aside, in funding and resource, this is something that I am struggling with on a daily basis. Certainly, the message that I'm receiving from sites and investigators is that the days where their goodwill is able to support an academic trial where they can put patients on or eat the costs locally are long gone. And one of the results of this is that we're seeing slower and smaller number of sites, slower activation and smaller number of sites that are participating on our trials. And that, of course, creates this catch-22 situation, which is if you can get a trial done relatively quickly, you are doing it at minimal cost. You're essentially saying that the costs that I have of staffing the trial can be amortized over a relatively short period of time, whereas if I have to staff a trial twice as long, it will cost me twice as much. So funding and resource, what do we do? Well, you can always try to fund yourself locally. That's really only feasible for a single center, perhaps phase 1 or phase 2 trial. It's not feasible for phase 3, in my opinion. The Canadian Institutes of Health Research is our go-to organization in Canada. It's a bit like the NIH for you in the United States. We are able to apply for peer-reviewed grants with them. The overall success rate is 10%. There's even a suggestion that there may be a bias against clinical trials in terms of funding them because they eat up the cost much more than, say, a basic biomedical science proposal. You could submit a proposal to a cooperative group like ourselves, and our investigators do that. We go through a process of scientifically reviewing those trials, but ultimately we also are at the mercy of needing to fund them. And then finally, again, you may submit a proposal to a pharmaceutical company or another company. Their support will be proportionate to their level of interest. There's always the question about investigator and sponsor independence. They tend to demand faster trials, more oversight, more demands. I know this is being recorded, Seth, and I know other people are going to see it. I have fabulous relationships with my pharma colleagues, but working with pharma is like being pecked to death by chickens. No single blow is fatal. It's just very, very irritating. And so you cannot underestimate the fact that if you are involved in a partnership or a collaboration with pharma, that you are answering, at least in part, to their needs and their demands as well. I'm going to conclude quickly with one final example of what I think was a very successful trial for CCTG, and then I'll open for questions. So this is a bit of shameless self-promotion. This was a trial that I was involved with in 2005. It was our CO17 trial. It took what was then a very novel drug, cetuximab, and we randomized patients to receive cetuximab and best supportive care versus best supportive care alone in a setting of refractory metastatic colorectal cancer. For those who don't know, cetuximab is a monoclonal antibody that binds the EGFR receptor and it stops downstream signaling. It had some early interesting and promising results. These were 3 trials that were done. The top trial and the bottom trial were single-arm trials of patients in the same population of refractory disease. The middle trial was a curious one where it was a 2-arm trial, a randomized phase 2 trial, but both arms of the trial received the experimental agent. And the bottom line was that there was some good early evidence. So there was a buzz. People knew what cetuximab was. Investigators had heard about it. They were aware of these results. They'd seen them presented at conferences. So there was a little bit of excitement that was coming when we announced that we were going to be doing this trial. We randomized, as I said, 572 patients in a 1-to-1 fashion. This was a bit of a struggle. The best supportive care was appropriate in the Canadian context. I'm aware, and we can talk a little bit about it in the questions, that running a best supportive care arm in the US setting may not be pragmatic. For us, it was something that we managed to do. We did this trial in collaboration with a group in Australia, and there the socialized medicine system also allowed us to have a best supportive care alone arm. Our primary objective was overall survival, and we were looking for a fairly modest improvement of a hazard ratio of 0.74. Between Canada and Australia, the trial recruited very well. We were very surprisingly quite accurate in terms of what we thought we would be able to recruit and what we actually recruited. And so over a period of 20 months, we recruited 572 patients. Most of that recruitment was across a number of centers. One of the things that I tend to see also in clinical trials is a handful of centers tend to provide the majority of recruitment. If you have breadth of interest and breadth of investigator enthusiasm, the more sites that you have that are putting on patients, obviously, the better that the trial can go. The trial was positive. It read out with a modest improvement, a hazard ratio of 0.77 with respect to overall survival. And we concluded that cetuximab was a new or should be considered a new standard of care in these patients. It was the first time that a single agent biological therapy had shown a survival benefit in colorectal cancer. It resulted in a New England Journal publication for us and our group. And the timeline, if I look back at it, although when I was living it at the time, it seemed quite protracted, it was actually remarkably efficient. From the time that we first discussed the trial to having a protocol ready to go, contracts signed, patients recruited, data in-house and matured, and presentation and publication, 5 years and 7 months is in today's world, this would be a very, very remarkable timeline to achieve. So was it a success? I think it met my criteria. I think it was well designed, properly conducted. It did give high quality data. We stringently analyzed it and reported it, and it changed practice. But why? Why did it work? Well, I think it was a relevant question. There was not another option for these patients. There was very promising data from other trials that created an enthusiasm and an excitement among investigators, and notably among patients as well. This is the new novel drug effect. It wasn't particularly simple, but it was required to be complex because the drug was given weekly. patients were willing to trade off the inconvenience of having a visit to the hospital every week for receiving a novel drug. Obviously, there were limited therapeutic options, as I've already discussed. It did not necessarily have a good benefit risk ratio from the perspective of patients. They had a 1 in 2 chance of being enrolled to a best supportive care arm, but overall, the trial ended up being something that patients did support, investigators did support, and in the end of the day, we actually managed to not only have a trial that was positive and change the indication for cetuximab use in refractory colorectal cancer, but we also had luckily collected those biospecimens that I referred to, and we were able to demonstrate that there was a very predictive effect of having a KRAS mutant gene in your tumor. And if you were wild type, you benefited, and if you were KRAS mutant, you didn't. And so the trial not only had that sort of initial impact, but went on to have other impact in terms of changing the indication to be in KRAS wild type patients. And in Canada, importantly, we had a health quality of life improvement, and the drug was considered cost effective within our single payer insurance system. So its metrics of success is that it's continued to live. But I think it as an example of a trial that at the time I perhaps didn't appreciate, but was something that was destined to be successful because it met all of those important characteristics that I referred to. So in the interest of time, I'm going to stop here, Sep, and open it up if there are questions.
Chris, I'm speechless. That was fantastic. And like really great metaphors. And I think what a beautiful success story to see. I do have a question for you, so I'm going to just start, and then obviously for anyone, please feel free to either unmute yourself and ask a question or just put it in the chat. We really want this to be interactive for the next like 7 to 8 minutes that we have before our second speaker. So Chris, I was curious. I mean, I think as you mentioned, like usually the trial gets approved and then really the hard work begins, right? And how, I guess, as a new investigator, what do you think are sort of, if you were to have like the little gravy of like what makes you the best investigator to make sure you stay up to date, like what are at least some key ingredients of how you keep your accrual and the timeline and everything?
So I think I'm going to be I'll deviate a little bit from what you've said. And I think what I would prefer to say is we have a community of young, upcoming new investigators, fellows, new appointed faculty, and they always ask the same question or a similar question, Sep, which is how do I generate a research career? How do I become a clinical trialist? How do I get my ideas on the table? How do I bring forward research proposals? And I always say the same thing to them, which is if you can work within an organization like ours, do so. And then if you're within that organization and you're an aspiring investigator, you're going to make yourself known by the support of the other trials that you undertake. So I say to young investigators, put patients on the trials that are available. Make the effort in your clinics to be a clinical trialist maybe it's not your trial, maybe it's somebody else's trial, maybe there are 2 or 3 trials that you're able to have open. But if you're putting patients on those trials, you will become known. And I can tell you absolutely that in our group, in the GI disease site, when we are looking for the next generation of someone to be a local PI or possibly on a trial committee, a steering committee, we are looking for the investigators who have supported other trials. So it's a self-fulfilling prophecy. If you can support others' clinical research, then you will be advancing your career and you'll be putting yourself in a position of having leadership or other contribution opportunities.
That's a great way of phrasing it. Akil, do you have any questions or anyone in the audience?
Yeah, Dr. O'Callaghan, I'll chime in here. So first of all, really fascinating talk and just great to hear your wealth of experience. And that last piece of advice you gave, I think is rings very true. So I think one of the important points that you had made was kind of focusing on the feasibility and cost of trials, and you showed those nice graphs. You kind of get a sense of how long some of these trials will go and the cost that's associated with that. And then the balancing point is that we want to get trials accrued to as quickly as possible. And here in the United States, we also have centers that really put patients on trials, and at times at the expense of not including underrepresented populations. And so, curious to hear your thoughts of how you balance heterogeneity or diversity in trial recruitment with simply getting a trial done.
Yeah. I mean, I'm going to say that one of drilling down on that specific idea of having equity of access to clinical trials. I have seen over the years a change. We used to write clinical trials with eligibility criteria, for example, that would say things like the patient must not live more than an hour from the enrolling institution. And in a country like Canada, we were uh, definitively discriminating against large proportions of our populations, and many of those were underrepresented groups. Our Indigenous peoples seldom live within an hour of an institution that's got a clinical trial running. And so, as an organization, we have tried to support broadening recruitment by moving to including as many remote procedures as we're able to make it as easy as possible for our patients to have things like telemedicine. Um, I hate to say it, but COVID changed the world for us because COVID made it suddenly essential to be able to look at patients on Zoom, to do Zoom interviews with patients, to follow patients remotely. And so we've learned lessons from that, and we're moving those lessons into our clinical trials. We're trying to have more and more of the routine follow-up be done in a way that is convenient for patients, so that when the patient is saying, well, I'm thinking about going on this trial, what does it mean for me? And the answer is, well, it doesn't mean that you're going to have to come in every 2 months. It means that you'll come in every 3 months, and the other visit will be done remotely by your physician. We have tried to do that. I can tell you that other groups have been more successful than us. Our colleagues in Australia have really done remarkable work, because the element that you have to balance there is the safety and the regulatory component. So you actually have to get your regulatory authorities to sort of understand and agree that this is the future of clinical trials, these decentralized clinical trials.
All great points. Any other questions from the audience?
Specifically, like, is there a reference, like a trial manual that
Yeah.
you recommend with common surgical oncology trials? I mean, I don't know about surgical oncology, but maybe you have some references.
I would have to think about that a little bit, Sep.
Yeah,
That I think it's very true to say, and this is not a derisory comment. The medical oncologists and the radiation oncologists are a bit ahead of you guys, but
100%.
the surgical oncologists are really picking up steam. And even within my group, 3 of the last 4 trials that we have been looking at to approve have been trials that have been led by surgeons. So I see that the world is changing a little bit, and the surgeons, I think, are picking up on the lessons that their medical oncology and radiation oncology colleagues have taught them. But I wouldn't think that there would be something specific to surgical trials that would be a reference manual. I mean, any opportunity to learn about clinical trial methodology and practical aspects of clinical trial conduct, don't look for it to be specific to surgeons. The principles are the same.
Yeah, I would agree. I have some references. Robert, if you send an email, I can, honestly, like I don't have like the full reference, but I certainly know of books that I read like during some of my training. So we can happy to share that with you. Well, it's six, unless there's any other questions in the audience, it's 6:45, so I'll turn it over to Akil, who will present our next speaker.
Thanks, Seb. So it's my pleasure to introduce our second speaker, Dr. Colin Weeks, who's an associate professor of medicine and the director of medical oncology research for pancreatic cancer at the Massachusetts General Hospital. In his role, Dr. Weeks focuses on clinical and translational research for patients with pancreas cancer with a specific emphasis on incorporating targeted therapies and biomarkers into clinical trial design and development. Dr. Weeks is also really involved in drug development for GI malignancies. And today his talk is going to focus on trial design, different approaches to answer your question within a clinical trial. He's going to provide valuable insights on how to design trials that effectively address various types of clinical questions. On a personal note, I've had the opportunity to work closely with Dr. Weeks during my surgical oncology fellowship training at the Massachusetts General Hospital. And at that time, his mentorship and guidance was extremely invaluable, particularly in shaping my approach to trial design at an early point in my career as I was developing my first trial. His insights and thoughtful leadership have only influenced me, not only influenced me, but countless other trainees and other faculty. And we're really excited to hear your perspectives today on this very important topic. Thanks so much for joining us, Colin.
Is ongoing. We've almost completed enrollment. Hopefully by the end of December, we will have completed enrollment to the study. We're seeing some initial sort of promising outcomes for our patients. So much so that then the next slide. So in this slide, as we all know, nab-paclitaxel has now been demonstrated to be better than, say, nab-paclitaxel and gemcitabine, at least in the NAPOLI-3 study. And so we've now began to engage with Ipsen to sort of see if we can do the next generation of this study, where we're going to combine the L-reclusive with nab-paclitaxel. So next slide. So I just want to talk about sort of how, we got funding for the study. And I think, as the previous speaker outlined, getting funding for your trials is, I think, both a curse and an opportunity. So we were fortunate in the initial study that I showed you to develop a partnership with Novartis, in which Novartis supported us during the trial. And then the specimens that we received from the patients were directly analyzed. They came out of the patient and went to the Novartis labs here in Cambridge, and they did RNA-seq on those studies. And so this was a really cool opportunity that we were able to develop this sort of really collaborative partnership. And I think, if the drug wasn't discontinued, I think it would have been really a partnership that would have lasted for probably most of my career going forward. The second study that I showed is supported by the Les Garden Foundation, and they have a clinical accelerator initiative. And I brought this particular study this foundation to your attention, in that I think one way to think about trial getting support for trials is through various foundations for the diseases in which we are treating patients. And so that is a way to, I think, one, get funding for your trial, but also, as was stated before, if you're doing trials and you're partnering with other institutions within these foundations, you will rise to the top just because you're participating in these situations. And then, as was said, it also said before, is that in the latter case with Ipsen now in the nab-paclitaxel study, we are getting support from Ipsen to do the next generation of this study. And I'm having to sort of change the study design based upon some input from Ipsen. And then I think it's also an opportunity for career development. And so the second study that I demonstrated, Dr. Priya Patak is a junior faculty member at MGH, and she's got a CDA from the WIN Foundation to do a clinical trial based upon this study. And I think it's also an opportunity for for junior faculty to sort of think about things like ASCA-YAs or other CDAs or other foundational CDAs. And then we've used both studies were part of either a PO1 or an RO1 proposal. So I think there are ways to use these trial designs to also develop a career and support your career. So last slide. So I'm just going to show you an approach that I've taken to sort of this question of diversification of patients on clinical trials. And so if we can go to the next slide and then just play the video that's embedded within that slide. Hopefully it works. Oh. Oh, unfortunately, so there's a video. If you go onto YouTube and put in FOLFIRINOX and NIS 793, this video will come up. And this video, what it does is it talks about localized pancreas cancer, metastatic pancreas cancer, and it's an animated video, and it talks about TGF-beta and how that modulates the tumor microenvironment. And then the last part of the video, it actually outlines the trial and using similar sort of diagrams as what I showed to outline the trial. But the idea here is that one, because you can see that the patients who are shown here are diverse patient population. If you were actually able to see the video, it would show so a cartoon of myself as a physician who's also doing the trial. So that would suggest to the people that individuals of color can be physicians as well. But most importantly, I think at the end of the day, this video, what it potentially does is it allows not only the patient, but the patient's family to sort of understand what the patient's going to be experiencing. And I think as, we commonly think about the patient who's in the room with us and sort of proposing the trial to just that patient. But really that decision is made amongst their entire family. And I think in more so in communities of color where the family becomes really important, and also getting some buy-in to what we're doing is going to be supported by the family. So this is a way in which I've sort of developed multiple videos of this nature for different trials to as a patient education platform to help with sort of clinical trial diversification. So I think I have one more slide, which is my last slide. And I can't do this without, all of my friends and colleagues, both at MGH and at other institutions across the country. So I'm sorry for all the rigmarole. So we have a few minutes for questions, but I'm happy to stay on longer too if people have additional questions.
Thanks so much, Dr. Weeks. I think you really nicely showed a prime example of a journey of how questions actually move from a single institution phase 2 study to then inform questions that we should be asking in phase 3 studies. Congratulations on all the amazing work that you and the MGH is doing in this disease space. I think you also showed importantly that these translational questions that you get answered from the initial hypotheses really come into play when engaging pharma and academic sponsors to support larger scale trials. When I think about short-term and long-term goals for proposed hypothesis, what comes to mind is the actual length of time that it will take to bring about the idea that you're posing to the actual scientific community. And because not everybody agrees with what your interest is per se, is there a way that you have identified in your clinical trial experience to move things along that line quicker? So, you outlined how endpoints, for example, R0 resection endpoint for a phase 2 single arm trial might be the definitive endpoint for that size of a study. But to make it practice changing, I think most people would like, progression-free survival or overall survival endpoint ideally. Are there ways that you've identified to quicken that process from inception of a hypothesis to getting it into prime time?
Yeah, so the second study that I showed, the metastatic disease study, there's actually a safety run-in component. So we're actually I didn't choose to show you the data, but we just we're developing a manuscript in which we're combining the outcomes from that safety run-in component in terms of survival and so forth with our preclinical work that supports the research. And I think what we're going to show in that manuscript is that instead of what's historically been done, where we just add drug X, Y, and Z to FOLFIRINOX, what we're going to show is that we're actually targeting a process that's being induced by FOLFIRINOX, right? So we're sort of targeting a pathway that is now really the cells depend upon this pathway in the context of FOLFIRINOX exposure, and that's where we're going to probably see some benefit. And we'll also show there's some pretty encouraging survival outcome in these patients, and so that way I think to sort of bring that buzz together, it takes time, right? And so by the time that this study gets it'll be another year or two before we actually publish the work on that study. But in the meantime, we're now able to design the next generation of that study, which will be now a larger study, right? So it is going to be a stepwise process, and thinking about in these earlier sort of study settings, I think thinking about shorter, more immediate endpoints allows you to publish that data sooner, right? So if we, in that first study, if it would have been able to complete, as soon as we did all the surgeries and we knew what the R0 resection rate was, we could publish that paper versus waiting for the survival data on that. And we could publish a second paper once we had the survival data down the road.
That's great. Anyone in the audience with any questions? Dr. Galami?
I mean, actually, that's a really good point, because I was just thinking, I would love to hear both Chris and both Colin's sort of thought on this. So this morning we presented actually an idea in which, we had a novel agent, think in the adjuvant setting for a specific patient population that obviously, as I'm talking, everybody knows what it is. It's, patient with liver metastases. But regardless, I think our proposal was something of a, I would say, small to intermediate type of study. And, with an early signal where, we don't have to wait forever and ever. And really what the feedback at the task force was actually just think bigger and go big or go home. And instead of just an initial thing and comparing it to historical controls and trying to, have something to work with, and then taking that as the ultimate randomized trial, and we were shocked. I guess it's kind of good news, but also it's how do you then think of, are we ready to then think bigger, right? And make the phase 2, 3, and think a lot bigger with obviously 2 different endpoints. How do you balance, trying to get the early signal and really, you don't want to invest too much, and too many resources and too much, in everything, finances, people, patients, but also get an answer to your sort of question. I'd love to hear both of you guys' input with your experience.
I mean, I think that's it's a good problem to have, I guess, in this context. And I think it also outlines, I think, I wouldn't say challenges, but one of the considerations when you work within a cooperative group, right, is that there are multiple eyes on a given situation with multiple experiences as it applies to that situation, right? And so, sometimes you do have to sort of amend your thought process based upon the feedback that you get, which is sort of being flexible is important, I think, in this conversation, because, you have your idea, you're in love with it, and someone comes along and says, well, maybe this isn't the greatest idea in the world. Let's do something a little bit different, right? And then you sort of have to sort of rethink. I think to the problem that you put in front of us, I think the way that I would think about that is I would design a phase 2, 3 study with sort of early stopping rules in phase 2 that are pretty stringent to make sure that when you do go into phase 3, you have robust data to support that. Because I think a lot of times what happens is in these type of situations, we don't apply very stringent rules for the phase 2 data. And so then we get into a phase 3 study and the studies don't tend to come out the way that we would have anticipated from these earlier phase studies. But Chris probably has much more experience than I do with these situations, so I'm going to defer to him a little bit here too. But that's my sort of 2 cents about it.
Yeah, I would say, Sep, that it sounds incredibly attractive and incredibly efficient to have a phase 2-3 design. But I think that sometimes the truth of the matter is that the strength of the academic cooperative group infrastructure is really for the phase 3 side, right? I mean, if you have got the data and you've got the foundational work done that would support a phase 3 design, it's sometimes cleaner and actually easier to move from a smaller focused phase 2, perhaps that you're doing alone or at your center, or you're doing with a select smaller group of collaborators, and then move that and sell that to the large cooperative group that's going to do the phase 3. I've pitched on multiple occasions, pitched phase 2-3 designs to pharma partners. And it's complex. It's very complex. And although I'm a great proponent of trying to, do things as quickly and efficiently as possible, I do think sometimes it's best if you get the knowledge and learn the lessons in a modest phase 2, smaller, more focused design, and then go forward with an idea to how are we going to take that into the phase 3 setting. The phase 2-3 designs are tricky.
Yeah,
Tricky to
Chris,
transition from one to
Chris,
the
can I
other.
ask you a question, a follow-up question to her question, which is, if you're a junior to sort of early mid-career faculty member, right, and you've got senior individuals or even task force leaders sort of saying you should do this, right? Um, and I agree with what you said, okay, but I can also see that Seppi's probably up against the wall in terms of
yeah,
these
I know
bigger
where the
pitch.
feedback came from.
Yeah.
Exactly.
Well, and
How do you navigate that, right? Because I think now this becomes sort of navigating the politics of a
yes.
cooperative group, not so much navigating the trial process.
Yeah, and you're absolutely right. That I think that there is an element of politics in that. There's an element of trying to gain that efficiency, right? So your perspective might be, or my perspective might be, let's keep this very discreet. Let's generate that short-term goal that you were talking about, Colin.
Yeah.
I want to here's my short-term goal to get out of this design, and then we can reconsider, we can regroup, we can plan. Whereas the leadership when you're in the task forces and you're in the steering committees, they don't want the small, they want the large, right? That's what they're kind of communicating to you why they exist, right? We're the big conglomerate academic group. We've got 5 cooperative groups in the United States that are here. We're not in the business of doing small phase 2 trials. We really want to do this and get the answer, change the practice. And so they're coming at it from the back end, and they're saying, okay, well, why don't you think about a phase 2-3 design instead of just doing your phase 2? I agree. It's hard to back away from that pressure, but sometimes it's actually better. I mean, I don't have an answer, Colin, when somebody puts you on the spot and says, why don't you do a phase 2-3? The answer is because I want to do the phase 2 and have a degree of certainty and confidence that what I'm going to have is going to be supportive of a phase 3, rather than get into a complicated situation where I'm trying to escalate quickly from a phase 2 to a phase 3 design.
Yeah, I mean, I think this is a really fascinating concept, the politics of cooperative group clinical trials, and maybe we have another session on this.
I know.
But
And
I
actually, I
really
was
want to
just
thank.
thinking about it. That might just be like a whole nother session on itself.
Yeah, I mean, sometimes the loudest voices are the ones that are most heard. But I really want to thank both of our speakers. I think this has been a really interactive session, and encourage folks on the audience to reach out if additional questions do come up. We will be sending out information with regards to our next session, which will be here in a few months. Thanks again to Dr. O'Callaghan and Dr. Weeks for joining us, and I hope everyone enjoys the upcoming holiday.
Yeah, thank you so much.
Thank
This was
you.
wonderful. Appreciate it.
Happy Thanksgiving, you guys.
Thank you. Bye.